Hi, how are you today? Now we'll start a new activity, which is activity 2, sexual reproduction and genetic diversity, formation of gametes, meiosis. Have you ever wondered why children do not look exactly like their parents, or why some siblings look so different? The answer lies in the process of meiosis, which generates the gametes for reproduction. Now, each cell contains 46 chromosomes in male and similarly for female. The sperm cell, if the sperm cell contains 46 chromosomes and ovum cell or egg cell contains 46 chromosomes, then after fertilization, we can obtain first cell of monster baby zygote which but with 92 chromosomes. Can this happen? In reality, each cell contains 46 chromosomes in the male, where initial somatic cell contains two unequal 46 chromosomes. Similarly, for female, where the initial somatic cell also contains two unequal 46 chromosomes, undergo a cellular division, which is called meiosis, in order to obtain sperm cell contains 23 chromosomes and ovum cell contains 23 chromosomes. This karyotype for sperm cell, which contains 23 chromosomes, 22 autosomes, and one sex chromosome, may be X or Y. On another hand, the karyotype of unfertilized egg cell also contains 23 chromosomes, 22 autosomes, and one sex chromosome, always X. Then, after fertilization, we can obtain a zygote, which is first cell of normal baby, but with 46 chromosomes. Then we can conclude meiosis solved the problem. How can we obtain such specialized cells with 23 chromosomes? In the previous section, we have discussed mitosis. The zygote, which is a single cell, undergoes the first division. I got what do we call a two-cell stage embryo. Two cells where each has 46 chromosomes, and this is the exact number of chromosomes in the zygote. And by successive division, the first division, the second division, and the third, the fourth, and the fifth division, after 5 successive mitotic divisions, we have obtained 32 cells out of one single cell. And if we look at this structure, which is E, represents 32 cell stage embryo where each cell having 46 chromosomes. This is a reality because of mitosis and due to the duplication of a chromosome that has preceded every mitotic division, and this is why in every two daughter cells the same number of chromosomes as the mother cell. Thus, the suggested answer to the posed question would be meiosis is another cellular division. Meiosis is a process of cellular division by which haploid cells, gametes, one unequal 23 chromosomes, are produced from diploid cell in order to make sexual reproduction. So, without meiosis, no fertilization and no reproduction. For this to occur, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells with half the number of chromosomes as the original cell. Sperm haploid cell, which contains an equal 23 chromosome, similar to the egg, which is also haploid and equal 23 chromosomes. What are the differences between somatic cells and sex cells? Some somatic cells, like muscle cell or skin cells, while the gametes are sperm cells or ova. There are two sets of chromosomes in somatic cells, while one set of chromosomes in gamete. On the other hand, somatic cell contains 46 chromosomes, 23 pairs of chromosome, while gametes contains only 23 chromosomes. In somatic cell, they have 44 chromosomes, in another word, 22 pairs of autosomes, and one pair of sex chromosome, XX or XY, while the gametes contains 22 autosomes and one sex chromosome, X or Y for sperm. Some sperm carries chromosome X and another one carries chromosome Y, and X for ovum. All the karyotypes of ovum contains a sex chromosome X. Meiosis occurs in special cells called germ cells within the gonads of males and females. These gonads are called testicles in males and ovaries in females. Testicles are the site where the male gametes sperm are produced by meiosis, while the ovaries are the site where the female gametes ova or eggs are produced also by meiosis. For the successful reduction in the number of chromosomes to occur in the new haploid daughter cells, two rounds of divisions are necessary. These divisions are termed meiosis 1 or reductional division and meiosis 2 or equational division. Although the processes of mitosis and meiosis look similar, there are few major differences. One difference is that mitosis results in two diploid daughter cells and meiosis results in four haploid cells. First, mother cells, which contains two unequal 46 chromosomes. Each chromosome exists in two copies, one from maternal origin and one from paternal origin. Moreover, each chromosome consists of two sister chromatids. After this mother cell undergo first meiotic division, which is called meiosis 1 or reductional division, consists of prophase 1, metaphase 1, anaphase 1, and telophase 1. In this phase, the homologous pair are separated, and we obtained at the end of first meiotic division two daughter cells. Each contains one unequal 23 chromosomes, which is called haploid. Then, these two cells continued the second meiotic division, which is called meiosis 2 or equational division. In this phase, similar to mitosis, where the sister chromatids are separate. In this phase, we have prophase 2, metaphase 2, anaphase 2, and telophase 2. At the end of this equational division, we obtained four haploid cells. Each contains one unequal 23 chromosome and each chromosome of one chromatid. As in mitosis, meiosis begins after a cell has successfully completed the G1, S, and G2, which is called interphase. During the S phase, of interphase, the DNA is replicated or duplicated, producing two copies of each chromosome called sister chromatids. Sister chromatids remain attached at the centromere and condense as the cell enters prophase 1 of meiosis. During prophase 1, pairing up of homologous chromosomes forming tetrads and become visible inside the nucleus. The paired centroids in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. During prophase 1 concludes with the fragmentation of the nuclear envelope. As the duplicated centroids pairs move to opposite poles of the cell, as they move, the centroids extend spindle fibers, forming the meiotic spindle. The meiotic spindle is fully formed, the sister chromatids attached to the spindle fiber. Now, during metaphase 1, homologous chromosomes form tetrads and line up to form the equatorial plate at the middle of the cell. Now, during anaphase 1, the homologous chromosomes separate and move toward opposite poles, which is called polar ascension. Meiosis 1 or reductional division ends with telophase 1, where the cell constricted at the middle and the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material and the two daughter cells are separated by a cleavage furrow. Since the final product of meiosis 1 is two haploid cells, meiosis 2 or equational division begins without the chromosomes going through another round of DNA replication or interphase. Centroids again duplicate and begin moving to opposite poles of each cell. In prophase 2, the sister chromatids condense while the spindle fibers start to form as the nuclear membrane disappears. During metaphase 2, each single chromosome lines up to form the equatorial plate. It's similar to the metaphase in mitosis. Now, during anaphase 2, two sister chromatids of each chromosome separate and migrate toward the opposite poles of the cell, called polar ascension. The entire process ends with telophase 2, where the cell constricted at the middle and the chromosome decondensed and the nuclear membrane reforms. Cytokinesis occurs and the cleavage furrow separate the two daughter cells into four haploid daughter cells. The haploid daughter cells will specialize into gamete, either sperm or egg. Fertilization is the process of union or fusion between the haploid male gamete, sperm cell, and the haploid female gamete, ovum or egg, to re-establish the diplody state and form a zygote, which will grow into a child. The child receives half its chromosomes from its mother, which is called maternal origin, and another half from its father, which is called paternal origin. What are the differences between meiosis 1 and meiosis 2? During prophase 1 of meiosis 1, homologous pairs of chromosomes appear condensed and forming tetrads, while during prophase 2, in meiosis 2, one set of chromosomes appear fully condensed. During metaphase 1 of meiosis 1, homologous pairs of chromosomes line up in the middle of the cell forming the equatorial plate, while in metaphase 2 of meiosis 2, chromosomes, each with two chromatids, line up in the middle of the cell, forming the equatorial plate. Now, during anaphase 1 of meiosis 1, homologous chromosomes, each with 2 chromatids, separate to opposite pole, while during anaphase 2 of meiosis 2, chromatids separate. During telophase 1 of meiosis 1, formation of 2 cells with half the number of chromosomes, each with 2 chromatids, okay, and moreover, the chromosome in the resulting cells is a chromosome of 2 chromatids, while in telophase 2, in meiosis 2, formation of 4 cells, each having the same number of chromosomes as the initial cell at the beginning of meiosis 2, okay, but the chromosomes in the resulting gametes is a chromosome with 1 chromatid. Because men and women produce millions of gametes, and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why each child isn't identical to either parent. But why aren't siblings identical? The diversity comes from several sources. In metaphase 1, the pairs of homologous chromosomes align independently along the metaphase plate and sort independently into the daughter cells. This process, called independent assortment, produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the power n. This means an organism with n equal 3 can produce 8 unique gametes. For a human, where n equal 23 pairs of chromosome, there are 2 to the power 23 unique gametes formed, or over a million different possible combinations. The combination of independent assortment and the random pairing of gametes during sexual reproduction increases genetic diversity and explains why a child will not look his or her parents or siblings. Finally, what are the differences between mitosis and meiosis? Mitosis takes place in all somatic cells or body cells, for example, skin, muscle, hair, while meiosis takes place in germ cells or sex cells, testicles in man and ovaries in woman. Mitosis favors growth and repair of injured cells, while meiosis favors sexual reproduction. One cell division only in mitosis, while in meiosis, 2 successive cell divisions, reductional then equational. During mitosis, the mother cell gives 2 daughter cells, while in meiosis, the mother cell gives 4 daughter cells. Mitosis preceded and followed by an interphase, while meiosis preceded by one single interphase. Finally, in mitosis, the obtained daughter cells are diploid to n, the same quantity of the chromosomic material as the mother cell, 46 in human, where the genetic information and the number of chromosomes are conserved, while in meiosis, the obtained daughter cells are haploid, 1n. It means half the quantity of chromosomic material of the mother cell, which is 23 in a human.